This is the 8th and final video presentation for the CASE Animal and Plant Biotechnology course. As you view this video, be sure to make notes of important information. Feel free to pause, repeat, and resume the video whenever necessary. Moving research into production, Lesson 5.2, from lab to production. You completed Project 5.2.2 Regulatory Roundup, during which you determined the influence of governmental regulatory agencies in biotechnology application safety. This presentation provides information about the steps and processes involved in bringing a new biotechnology application to fruition. Moving a new discovery from research into production requires a great deal of time, testing, and trials. There are many phases to test the reliability of the products and the possible effects it may have when produced. Critical stages include manufacturing, risk analysis, regulation, and testing. Good manufacturing practices are a set of standards that are enforced by the FDA. GMPs ensure that safe practices are used throughout the production process and that all materials are of high quality grade. Workers must follow proper laboratory practices, facilities must be maintained, and formal processes for developing and testing must be followed. All products must be demonstrated to be of consistent quality and form. Risk analysis is used to determine how safe a new technology is and how it should be monitored. All risks are weighed, measured, and evaluated against the potential benefits of the technology. Questions and considerations for a risk analysis differ based on the organism being modified and the product desired. Considerations when conducting a risk analysis include resistance, negative characteristics, target species, impacts, health effects, non-target species impacts, and potential for transfer. Potential threats or risks are categorized according to many factors. Products with low levels of transferability and reversible effects require less monitoring than those with high levels of transferability and irreversible effects. The index of risk takes into account the damage, exposure, and precedent. Damage is the level or intensity of the impact that the GMP could have on the system, if the proposed adverse effect actually occurs. Exposure related to the level that some component, for example soil, animal, native plant, is exposed to the damage. The higher the exposure, the higher the possibility of an adverse effect occurring. Precedent considers the previous occurrence of the adverse effect as a consequence of the event in question. The index of significance takes into account the extent and reversibility and includes the location where the GMP will be cultivated, the identification and evaluation of the potential adverse effects, and the evaluation of the current environmental situation. Extent reflects the extent of dispersal or the distribution of the damage. Reversibility is the ability of the system to return to the previous condition. Regulatory agencies studied in the previous activity play big roles in the process involved in bringing new biotechnologies to the marketplace. Products must undergo extensive testing in order to determine the reliability of the results and all potential effects, side effects, and other outcomes. If lab testing has positive results, the product may move into limited field testing. As a review, mark or highlight 3 key points in your notes that are important to remember from this presentation. List 2 ideas or concepts that relate to previous knowledge. List any questions you have about this topic. Discuss these questions with your instructor. Keep any notes you have from this presentation organized and available for use throughout the course. Through Project 5.2.3, Making Research a Reality, you will develop a model depicting the steps from laboratory research through production of end use of the biotechnology. This will help you demonstrate how results of research have to undergo multiple steps and trials before reaching consumers. Your teacher will provide more instructions and expectations for the product, along with Project 5.2.3 evaluation rubric.